All right. Well, first of all, I want to thank the organizers, the Gibraltar National Museum, for inviting me here. It is always such a great pleasure for me to be here in Gibraltar, not only because of the incredible hospitality of the museum staff and of the city, but also because of the incredible engagement of the people of Gibraltar in these conferences and these meetings. It is one of my favorite places to come because you have just incredible people who come out for meetings who are so interested and engaged in our science that we do, but in our heritage and where we've come from. And in my talk today, I want to engage a bit with some of that heritage in a way that is new for me, and I think will probably be new for many of you. I also want to welcome the people who are watching on YouTube. If you're watching, I think it's wonderful that we have live streaming of these meetings, and I think it's great that we're able to reach out beyond Gibraltar to the rest of the world. I know that I have many, many friends who watch these things, and I just love that we can all come together and still be reaching out beyond the boundaries of the room. I want to talk today about the ancestors of the Neanderthals. I want to lead off by saying I do not know who the ancestors of the Neanderthals are. And I don't think that any of us have a great idea of who the ancestors of the Neanderthals are, other than what I will tell you, which is that Neanderthals had many ancestors. We can point to a few of them, but the formation of the Neanderthal population, that deep time perspective on this population that we know so well from the fossil and archaeological records, but we know relatively little about its earliest stages and the other populations that existed at the time they originated, it right now is a very engaging mystery. And I'm going to go into some of the reasons why this is today mysterious, where the Neanderthals came from, who their ancestors are. I'm going to talk to some extent about the genetic record, and I'm glad Svante's not in the room so that I can say whatever I want. And I'm going to try to align what we're appreciating from the genetics with what we can see from the fossil record, little as it may be in many cases. And I'm going to point out what we don't know about the accuracy of genetic models and about the sort of accuracy of fossil observations that makes this a compelling problem right now. So that's where I want to get to in this talk. The end of the talk is, we don't know. So if you expect knowledge, you can leave now. All right. So I want to begin by saying, one point that's going to go throughout, and I put this on here to remind myself, confusion about where the Neanderthals came from is in large part due to confusion about what Neanderthals are. And this is an issue that's remained with us since the discovery of the Neanderthals. And I'm going to highlight some of that history, but I want you to sort of retain that. Confusion about where they come from is intrinsic to the fact that we're confused about what they are. All right, so first I want to talk about briefly, we are learning incredible things about genetics, right? This last couple of weeks, we have the news of an individual who has a Neanderthal parent and a Denisovan parent, right? Neanderthal mother, Denisovan father. And this is a very special individual from Denisova cave, and the genetics tells us about this. Now, the paper doesn't use the word hybrid, ever. You do a full text search, right? There's no mention of the word hybrid. The cover of Nature says hybrid. So clearly, there's a mismatch of communication between what the geneticists and what the genetics is telling us and the way that this is being portrayed to the public, right? And that's something to note, right? When you have the cover of Nature that says something different from the paper that the cover is representing, it's telling you there's something mysterious about this. I'm going to try to highlight a bit of that. You can, ask other people who also know about the genetics about it. But I want to focus, to begin, on one thing. The discovery of these ancient populations from genetics has been transformative for our field. We're finding populations by sequencing DNA from ancient fragments of bone. We're finding populations that archaeologists and anthropologists had not guessed existed. What does that mean? Because we guess at a lot of things, right? And when we guess too much, we get criticized. People say that's X-Files paleontology and this sort of thing, right? So it's we're making stuff up. So there's a point to which you have to be very conservative in what you're willing to say. I want to highlight a case where genetic knowledge leads to this sort of expanding cone of sort of ignorance, all right? So let me just start with, this is a map, right? This is from Nature, from a couple of years ago. And you see here, we've got the map that shows the geographic range of Neanderthals, which is sort of blue. The gray is modern humans who spread from Africa 200,000 years ago around the world by 50,000 years ago. And the hatched places are the places where they overlap. And this pink glowing glob is Denisova Cave, where you have this other population. And Denisova becomes super interesting because a component of the genetics of the individual found at Denisova Cave is found in Australia and New Guinea, so there's a contribution from this population to today's populations, and yet it's not Neanderthal, so it's something else. This encompasses a fairly accurate view of knowledge, I would say, in about 2011, 2012. Here's the range of Neanderthals, here's Denisova. Now, I want to point out the range of Neanderthals, right, where Neanderthals lived. This is rather a nebulous concept. Where did Neanderthals live? Those of us who teach this stuff can answer the question, right? Neanderthals are the sort of middle and early late Pleistocene population of Europe, and we recognize that there's these other places that there were things that people call Neanderthals, the Levant, for instance, Iraq, for instance, Uzbekistan, for instance, which is way over here, the Altai, for instance. When you find specimens that you attribute to this population, it expands the range, right? It's what Milford said about the earliest a little bit ago. There's always an earlier. Well, when you find something outside the range, it expands the range. There's always a bigger range. Now, these ranges have a way of mysteriously expanding. And this is from New Scientist, covering the same research that Nature was covering with the other map, right? And here you've got the Neanderthals, including the Altai specimen, which is from Denisova Cave. They call it the Altai specimen because to retain confusion, that it's from the same place as the Denisova specimen. In fact, all of the Denisovan specimens are from this one place, and a Neanderthal specimen is from this place, and the hybrid specimen is from this place. It's a wonderful place, right? It's a very special place. It's a great cave to visit. I recommend it to everybody. But you see that, well, the fact that we've got their genetics down here means that there must be some sort of Denisovan range, right? They must have lived somewhere. They didn't just live in this cave. If they did, they had to sort of vacate it at the time the Neanderthals moved in. So there was some kind of a, trade-off. And so they have to be somewhere, and here they are, right? It's the north-south realm and the east-west realm, okay? Now, this is all popular science, right? So I'm not, but I will say that the popular science reflects a view of where the Denisovans lived that is held by professionals. All right, now we've got the Neanderthal sort of Denisovan frontier, and here it's, sort of at the Pakistan-India border, it looks like. And you've got these Neanderthals, they're in the range, and then you've got these Denisovans in the range. Well, wait a minute, the only Denisovan that exists, of course, is up here. So these ought to be Denisovans, right? And you ask our colleagues, and they will tell you, I know that one of these is a Denisovan. We're going to find the DNA. It could be, right? I'm not discounting the fact that we may discover that the DNA from a specimen from this area might share the same phylogeny that the Denisovan individual shares, and so as a consequence, but you can see what will expand the range. This one's better because you've got the Neanderthal range. The Neanderthals are really pushing on the Denisovans. But the problem is that you've got this specimen from Western China, from sort of west-southern China now, that has Neanderthal traits. And so as a consequence, the Neanderthal range has to push this way, and it shrinks the Denisovan range. And then you've got erectus. Now, erectus, we have no positive evidence of erectus existing anywhere in the world after 450,000 years ago or so, right? We talk about them existing later, as if this part of the world must be erectus realm, and here's Denisovan realm, and here's Neanderthal realm. And notice, right, the fact is that all of these maps are built on a fundamental assumption about the way that hominin populations behave. They behave like empires, right? Now, they push their frontiers, they push their boundaries, they defend a range. Now, this is not out of fiction, right? Because when you look at the distribution of mammal species today and mammal subspecies, you find that there are zones where populations come into contact, where species come into contact. There are sometimes hybrid zones, right? There is a sense of geographical unity that exists within animal populations, and we can expect that hominins had some sort of geographic unity. Right? In some sense, they don't really behave like empires, which, as we all know, the boundaries of the largest empire ever to existed in the world were one giant contiguous block, which included Gibraltar, correct? No, it was not a contiguous, unless you count the 7 seas, right, which makes it contiguous. If Britain rules the waves, then I suppose it's all contiguous. But it's not contiguous, right? Humans do not behave as if their geographic ranges are contiguous, even though when you look at humans as animals, you'll see that populations tend to have some sort of geographic scope. The fact is that when we find different populations in one place, over 100,000 years or something, as we've done in Denisova Cave, with something that looks Neanderthal-ish in its genetics and something that looks very different in its genetics, we can expect that there were multiple populations that existed at different times that moved some large spaces, and there were probably significant times when there was almost no occupation, maybe total absence of humans. And the fact is that humans are fluid in some sense across the landscape. So I want to go to what the fossil evidence is. So to start with, what is genetics telling us? I'm going to come back to genetics in a second. But I want to first dive into history a little bit. The fossil evidence of human evolution is spectacular, right? If we compare it to other kinds of creatures, compared to the feathered dinosaurs, we have tons and tons and tons of human ancestors and relatives represented in the fossil record. But it is a very biased record. It's a record that is biased in crania, teeth, and mandibles, right? This is a wonderful array at the Smithsonian Institution that shows you can look at skulls from every time and place in human evolution. But if you want to look at skeletons, skip the Neanderthals and Cro-Magnons, except to say, our understanding of Neanderthals comes from this time, more or less, right? It comes from the time that we cared a lot about skulls and mandibles and teeth, and a little bit less about the rest of the skeleton. I'll tell you that when it comes to the rest of the skeleton, and this is the figure that Milford showed you a little bit ago, we have only a few specimens from the first 95% of the hominin fossil record that show you skull, mandible, teeth in association with postcranial evidence, right? Here they are, some of them. And you can see that they're scrappy. There's not very many of them. With Neanderthals, we have a luxury, which is incredible in our field, that we have much of the skeleton represented in many individuals from some extraordinary sites, right? This is Krapina, here's Jakov Radicic, and you've got incredible array of skeletal evidence from Krapina, right? 120,000 years old, more or less. From Cima de los Huesos, which I'll discuss later as an early member of the Neanderthal lineage, you have more fossil evidence than you have from any other site of hominids, right? You have an incredible array of evidence from this site. You've got cool skeletons like this Homo floresiensis skeleton, right, from Liang-Bois. So we've got new discoveries that add skeletal evidence later in the human fossil record. We've got our site from the Rising Star Cave, the Homo naledi material, which is, again, a large array of fossils from around 250,000 years ago. Again, we're looking at the last 5%, more or less, of our evolutionary history, but we have a depth of evidence that we don't have in earlier time periods. And yet, nonetheless, this is the Denisova finger bone that generated the genome, that tells us there's a whole population, right? This is it. And yet, one tiny fragment can change the way we look at things. And that's a humbling message, right, as a scientist, because what you're saying is, we've got all of this evidence that covers the last 5% of our evolutionary history. We know a lot about this, and yet I can find one finger bone, and it could totally change things. Right? This really cool specimen from Mislia Cave that was published earlier this year, that we have Mina in the room who's responsible for this. It's a wonderful, wonderful specimen that changes our view of the geographic distribution, the dispersal of human populations, right? This aligned with early African populations in its anatomy. Maybe this is the earliest modern human outside of Africa, et cetera, et cetera. All I'm saying is that even though we have a lot of evidence, nonetheless, we still act as if, for good reasons, the fragments shape the way we think. And that's maybe humbling, and it maybe should make you sort of cautious. Well, in the olden days of the 1950s, they didn't have nearly as much evidence. I'm not going to go back to the beginning of Neanderthals. There are other people in the room who can talk about the early history much better than I can. And I'm not especially going to talk about what this chart from F. Clark Howell is responding to. But I'll mention, in the days when Clark Howell began writing about Neanderthals and their variation, and to my mind, some of the first modern thinking about where Neanderthals came from, he was responding to an older idea that Neanderthals had existed in parallel with our species, Homo sapiens, throughout a long, long period of existence in the lower and middle Pleistocene. This became known in the 1940s and 50s as the pre-sapiens theory, the idea that there was very, very early modern human-looking population that had existed in parallel with Neanderthals. Howell was responding to that by saying, look, the evidence for this, he uses the word downright flimsy. I don't think we can get away with publishing this anymore about people. There's downright flimsy evidence that there's some deep, deep lineage of modern humans. He says, if you look carefully at Neanderthals, you will discover that there's much more complexity than people have been giving this credit for. And Howell raises the issue that I started the lecture with. Howell raises the issue that there's confusion about what Neanderthals are. Howell tried to address this confusion by delineating different kinds of Neanderthals. He says there are classic Neanderthals. Those are the Western European things, like La Chapelle-aux-Saints, like La Pharisee, like the Gibraltar skull. These are classic Neanderthals, and the classic Neanderthals represent an anatomical extreme that is not closely connected to modern people anywhere in the world and has its own sort of European identity. But when you look at earlier Neanderthals, like the Krapina sample, like the sample from Saccopistori, what you see is less development of these very specialized looking traits and more sharing with modern humans. So that these early Neanderthals, here he places them here, these early Neanderthals are a stem population, potentially, for both the classic Neanderthals and some modern human population. And so he says, where you have Neanderthals originating is in this phase where you might well have early Neanderthals in some sort of evolutionary stage point of view that are represented in other parts of the world where we haven't found them yet. And some of those populations gave rise to us, modern humans, through a process of sapienization. And we see the evidence of that manifested in the Levant, in sites like School and Qesem, and we see hybridization between School and Qesem and more Neanderthal-looking people, which gives rise to things in his day, like Tabun. And as a consequence, you have this sort of late origin of Neanderthals. Right? And he points to evidence of this. He shows how you have in classic Neanderthals, you have this very straight malar border here. And in an early Neanderthal like Saccopistori, it's more human-like in its anatomy. He shows by sagittal cranial outlines, right? Here, Saccopistori is the dotted line here, which has this sort of less extreme elongation than La Chapelle-aux-Saints and Montecerchio, right? Two classic Neanderthals. He shows early Neanderthals are not like the classic Neanderthals, and early Neanderthals are a potential intermediate in some way and maybe represent an ancestral population to both, right? Let me also say, He also looks at the Levantine populations, and the Amud skull was not available, I think, to him at that time, but I'm using it here as a stand-in because you see that here's Amud compared to School 5, which are not the same, but share quite a lot of similar morphology. Here's the Amud skull compared to Sacapastori, which is an early Neanderthal in Howell's point of view. His point of view on Levantine Neanderthals was that they also were a less extreme population, possibly the result of hybridization between early Neanderthals and modern humans. Okay, so that was Howell's point of view. This point of view, I think you can see today, is very much like, it's not identical to, but it's very much like the point of view that we've heard from genetics in the last 10 years. You hear people writing about the genetics of Neanderthals and Neanderthal origins. They, 10 years ago, 8 years ago, were writing about a relatively late origin of Neanderthals. Neanderthals diverging from modern human lineages, the African ancestors of modern humans in that point of view, something like 250,000 years ago. That early Neanderthals, like Krapina, would be after that time, but they would represent a less extreme version of the Neanderthals that ultimately would come to exist in Europe. And maybe some of these earlier, slightly populations are common ancestors of modern humans and Neanderthals. Something like the Simondolus huesos, right, sample would be something like a common ancestor. That was a very common way of talking about the genetics, something like 8 years ago. All right. Oh yeah, here's Zuttiyeh. Now maybe something like 250,000 years old, and something that Howell would put in the ancestral population. Today, now I want to talk about phylogenetics. You saw some trees in the last lecture. I'm going to show you a couple of trees in this lecture, and I don't want you to take any trees seriously. The dinosaur tree, take seriously. That's 100% the work of really serious people. The hominin trees, I don't know. The ones that are the work of serious people, maybe you shouldn't believe. So here's a tree. And this tree has the major defect that there's a couple of things on it that I want to talk about in a while that aren't on it. But it has the major advantage that I've put question marks in a lot of places because we really don't know. I'm going to talk about the question marks in just a second. But here on this tree, just to orient you, we have Neanderthals on the tree. We've got archaic humans in Africa on the tree. We've got modern humans, today's humans on the tree. We've got Homo erectus on the tree, and Homo floresiensis is on the tree, and Homo naledi is on the tree. And they're more or less in time sequence, right? The deeper in the tree you go, the older you get. It's hard by putting blobs that represent skulls on a tree to get time precise. So I haven't tried. But you've got also these robust Australopithecines that Milford talked about in his last talk. You've got these earlier sort of species that are attributed to Homo here. You've got Africanus here. Okay, so Milford's point of view, these guys would all be Homo. In other people's point of views, maybe these guys are Homo, or maybe these guys are Homo. Right? And why is this here instead of being in Homo, right? We are divided about where to divide Homo from the rest. I'm not going to go into that. I want to focus on these last parts of the tree. And I'm going to show you a couple of cladograms now. These are the results of a recent study that our team did on Homo naledi. And so Homo naledi is pink here. And I left it pink so that I could show you how its position changes. This is a tree that's based on looking at skulls and teeth of all of these different forms. And the skulls and teeth are not the whole skeleton, but the skulls and teeth are all that exist of some of these forms, right? You guys saw Homo habilis is almost entirely represented by skulls and teeth. Rudolfensis, there's no postcranial material that anybody's ever called Rudolfensis. Some people would not call anything Rudolfensis, right? So there's some uncertainty in what goes into this. What I want to point out here, this is a parsimony tree. This is a tree that you would get, if those of you who've done parsimony analyses in 1995, if you took the characters from this stuff and put it into PAUP and plugged it out and drew the results in MacClade, right, you would get this tree. This is a parsimony tree. And the parsimony tree has humans today, modern humans, and Homo heidelbergensis as sister groups. Neanderthals as an outgroup to those two. So humans are connected to heidelbergensis. It's got erectus all together here, and it's got some early forms of Homo together here with Kenyanthropus, which is that sample from Kenya, from Lomekwi that you saw. And Homo naledi is way out here as sort of an outgroup to the rest of Homo, except for the hobbits, except for Floresiensis. Okay. Now, the entire reason for going through this is that the next tree that I'm going to show you is totally different. This tree is based on the same data set, entirely the same data. There's no difference in the data. The difference in this tree is that they've used a Bayesian model instead of a parsimony model. And the difference between those will be important to maybe one person in the room, but most of you, the only thing that I want to point out about this is here you've got, well, Neanderthals and heidelbergensis are sister groups, and modern humans are out to them. And then you've got Homo antecessor connected to them. And then naledi is actually with these derived humans, the big-brained ones. And you've got erectus is more distant, and then you've got Floresiensis. Look at this. Here's a species that's completely changed its position in the tree, right? What does this tell us? It tells us that this species, by the way, is the one where there's all the data, right? So lots of data for this, lots of data for Neanderthals, lots of data for modern humans, lots of data for erectus, honestly. A pretty good amount of data for Floresiensis, for that matter. The fact is that even for the best fossil samples that we have, the way that we analyze the tree gives us totally different trees. This is a hint that the data on which of these fossils are related to each other are not very convincing. That's where I want to go with this. I want to just point out, the fossil evidence does not tell us with much resolution which species are close relatives of each other. Now, these trees, those two trees, share a couple of features, right? The big-brained humans, Neanderthals, and heidelbergensis are grouped together in both trees. That's probably genuine. If you asked me, what are the closest relatives of Neanderthals among all the hominins, I would say, well, probably today's modern humans and other archaic humans that have big brains are probably the close relatives, right? This is maybe not a surprise. But the fact is that we can't say anything more than that. The phylogenetic approach doesn't tell us who the ancestors of Neanderthals are. All of these confidences in all of these, right, this is the one that has all the confidence, the one that groups those three. That's why I say it's sort of replicable. The others are sort of miserable. The other ones are not replicable. A slight difference in the data would result in a totally different tree. A slight difference in the method of analysis results in a totally different tree. So as a consequence, the anatomical data that we have from Neanderthals, from modern humans, from African archaic humans, from Homo naledi, from everybody, is not good enough to tell who's related to whom. That really sucks, right? This is where the geneticists come in, right? Because the geneticists would say, we know who's related to whom. We've done their DNA. It's just like Ancestry.com for hominids, right? We know who's related to whom. And here's a tree that represents 2015 knowledge of the genetics of Neanderthals, Denisovans, modern humans, and other things, right? And here's the salient feature of it. We have today's people all on one branch of this great genetic tree. We have a second branch of this great tree that has two big populations on it. All of the Neanderthals that we know about are on one of them. And all of the sequences that classify as Denisovans are on the other one. And in addition to that, we have evidence from genetics of mixture among these branches, right? So the Neanderthals contribute to, here's Neanderthals, contribute to the ancestors of all these populations who don't live in Africa today. And the Denisovans, somebody, not this sequence that we actually have, but a mystery sequence that is a sister group of that one that we actually have, contributed something to Australian and New Guinea and so on populations. And that branch has a big input from somebody that we haven't found yet. This is the ghost population, right, that diverged at the very base of this tree and contributed to Denisovans, but not to Neanderthals. Okay, well, I believe in ghost populations. I do, right? I see ghosts. And I'm going to talk about more ghosts. But I will say at the outset, the ghosts are a consequence of a model. And we should be aware and cautious when the model generates ghosts, right? Because the beauty of a model generating ghosts is it gives you something to predict, right? I predict there will be ghosts. I predict that there were other populations that were quite genetically diverged that contributed to these populations. That's a prediction. And that prediction can be tested by finding them, right? And that's wonderful. We like that. But there are possibly other explanations that might diminish the importance of these ghosts or make them disappear entirely in some cases. And that's something to keep in mind, that there are assumptions that underlie these models. Okay, now, the first thing I want to say before I go terribly far into this is that humans are not weird. Humans are weird in some ways. But in this particular way, the fact that there are deep branches, genetically quite divergent from each other, that mix with each other sometime during the Pleistocene, this is not weird. This is the tree of chimpanzees using the same methods, right? Here's bonobos, here's Central, Eastern, Western chimps, and a population that is sort of geographically intermediate between East and West chimps. And what I want to show you is that these branches go back in time among the chimpanzee subspecies today. Here's 139 to 182,000 years between Eastern and Central chimps. Here's 250,000 years between Western and this other branch of chimps. Here's 500 to 600,000 years between those two big chimp divisions. And something like 1.5 to 2 million years between chimps and bonobos. And after that time, all of these branches have some hybridization among them. So the fact is that this is actually the universal pattern that we know of for mammals. Every mammal lineage that has been examined so far with contemporary DNA or ancient DNA, including extinct ones like mammoths and the cave bears, right? Earlier this year, we discovered the cave bears contribute to brown bears, the brown bears mix with cave bears. Those of you who are archaeologists who have to worry about the cave bears and brown bears understand there are hybrids in your future. They're coming because the fact is that these guys all mix with each other rarely. And that's the other side of this picture, that mammal species do have these deep lineages within them that are genetically they had time to become different from each other, that interbreed with each other, and interbreed with each other on the order of something like 3% or 2% or 7 to the minus fifth power percent. That's not very much, but the 16% is the highest here, right? So yeah, they interbreed with each other on a percentage basis. So hominids, the tree is not weird, right? The tree is actually perfectly ordinary. What's informative about the tree is the divisions that we have found, but the divisions that we've found are not necessarily the divisions that existed. And these ghost lineages, I'm going to talk about a couple more of them in a second, give you hints of that they're divisions we haven't found yet. Okay, now, some things that we know about Neanderthal variability from genetics today. First, here's the Denisovan. This is a PC plot, and there's nothing particularly important about it except that you see that the biggest difference among these sequences, the PC1 difference, is the one that separates Denisova from all the things that we call Neanderthals. This is why the geneticists call them Neanderthals. The geneticists do not call this specimen a Neanderthal because of its anatomy. It is a toe phalanx. Right? The geneticists call this a Neanderthal because of its gene sequence. If you set it up with Denisovans and other Neanderthals, it consistently looks more like the other Neanderthals. But when you look at the second component of variability among the sequences, what you'll see is that all the rest of the Neanderthals sort of look a lot more like each other, and this one looks really different. So the fact is that Neanderthals, as a category being used today by geneticists, and I'll blame the geneticists for this, because the fact is the anatomists did not introduce this problem of toe classification, right? The anatomist would not have said, oh my goodness, this toe bone is a Neanderthal. Now, you and I both, all of us know an anatomist that would say that. And so I don't want to go overboard, right? There are some. But the toe classification is not very effective. The genetic classification could be effective, but note that this sequence is actually quite different from the others. And I want to talk about that for a second. It's been known now, this is a 2012 paper from Levantine. It's been known now for a while that Neanderthals encompass more mitochondrial DNA variation than living people do. Now, I want to be really precise about that because you'll hear other people say the Neanderthals are much less variable than living people are. Both of these things are true. But the way that we measure variability differs in different contexts, right? If we look purely at mitochondrial DNA sequence divergence, we look at later Neanderthals in Western Europe, and they're all they look like some very tiny human population in terms of their variability. But if we broaden that and look at earlier Neanderthals and Neanderthals that live in other parts of Western Asia, you find that they're much more variable. If we look at the nuclear genetics, we get a similar picture, right? We have later Neanderthals, largely in Western Europe, that are all close to each other. Then you've got some Western Asian, I guess you would call Ukraine very Eastern European, sequences that are a little bit more different from that. And then you've got this Altai sequence, which is actually really different. If we broaden this to look at the mitochondrial picture, which has many more samples, right? It's narrowing in terms of what we're looking at, but it's broadening in terms of representation. What you discover is that, oh yeah, this Western European sample is all really, really similar. That encompasses less variation than today's Gibraltar population, for sure, in mitochondrial terms. But if you broaden that and look at other Neanderthals from earlier in time and from other places, you see, oh, they're much more variable. And when you look at this Altai sequence and the Hohenstein-Stadl sequence, you say, whoa, there are these really divergent branches of Neanderthals out there. And then when you look at Cima de los Huesos, which in nuclear DNA terms, was it on that last chart? No. But if you look at it in nuclear DNA terms, it branches with the Neanderthals, it doesn't branch with the Denisovans, it's got a totally different mitochondrial sequence. Its mitochondrial sequence is closer to Denisovans, but it's not close really to anything. You've got a complete replacement of mitochondrial DNA, as far as we know, in Europe and Western Asia among the Neanderthals. In other words, we look at Neanderthals, we are interested as scientists, as anthropologists, we talk about Neanderthals as if we're talking about this empire that existed from the Altai to Gibraltar and was contiguous over space and represents populations that were forming a race, right? They were becoming really, really different. The fact is that that empire was full of different populations that lived at different times that were actually much more different from each other than any living people in that region are from each other. They're comparably different from each other as any living people in the world are from each other, right? From Qesem to Beijing, that difference in genetic terms is the difference that we have among Neanderthals in genetic terms. So it's sort of crazy level difference. And that difference has gone through a succession over time with mixture and input in significant ways from other populations. So this chart from a 2016 paper was one that demonstrated that there was a stem population of modern humans that contributed DNA into this Altai Neanderthal population, but not the others. Again, this estimation comes from a series of assumptions and models. I don't want to take it too seriously. What I want to say is that it's not true that Neanderthals didn't get DNA from us. In fact, Neanderthals got DNA from modern African humans multiple times during their evolutionary history. Not only in the later phases, right? This is estimated something like 100,000 years ago. So you imagine those Skule and Kafsa critters running around and sending their DNA into the Altai. That's the time frame we're talking about. But remember, the Cima de los Huesos sample has totally different mitochondrial DNA from any later Neanderthals. How did that happen? How did it come to be that today's people are the closest sisters of Neanderthals in their mitochondrial sequence when when Denisovans are the closest sisters of Neanderthals in their nuclear DNA sequence. In fact, we know how this happened. The reason why, up until 3 or 4 years ago, the geneticists were talking about a recent origin of Neanderthals was because this deep data set of mitochondrial DNA shows that Neanderthals are close to modern humans. And the only way that can happen is if the mitochondrial DNA had branched recently between Neanderthals and modern humans, something like 400,000 years ago. So as a consequence, the idea was that Neanderthals and modern humans went their separate ways 200, 250,000 years ago, and we had this recent Neanderthal origin. Today we appreciate that genetics of the nuclear DNA tell us that, no, the origin was much older, and it involved this Denisovan population, which is really separate. It has to have been deep. The only way to square that circle, to reconcile those two observations, is for Neanderthals to get their mitochondrial DNA from African modern humans, or for African modern humans to get their mitochondrial DNA from Neanderthals. One way or the other. We don't know which. So there's a deep episode of gene flow in the origin of Neanderthals from African populations or to African populations that occurred sometime before 300,000 years ago. So you've got this history of successive population mixtures which have contributed in a meaningful way to what we today can see of the genetics of Neanderthals, right? I'm not just talking about trivia. This was the data on Neanderthals before 2010, the mitochondrial DNA. Everything that anybody wrote about them genetically before that was based on this, and this is an introgression that came from elsewhere. Well, okay, that's pretty cool, right? That's a really exciting observation that we've got this deep mixture and that it contributed to what we understand of Neanderthals. It is a way of saying that among the ancestors of Neanderthals are African populations from the Middle Pleistocene. That's important, right? African populations of the Middle Pleistocene are among the ancestors of Neanderthals, in the same way that Neanderthals are among our ancestors. It's precisely analogous. We've got things from Neanderthals because they're our ancestors. It is an unfortunate thing that anthropologists everywhere must train themselves to talk about our African ancestors and our Neanderthal ancestors. It cannot be modern humans and Neanderthals. That's not a meaningful concept in genetic terms. It is our African ancestors and our Neanderthal ancestors. And potentially our Denisovan ancestors, right? Our potentially other ancestors. So let's look more deeply into this. Alan Rogers last year did a really cool model about Neanderthal origins and Denisovan origins, and the really cool model was accompanied by no useful figure. This is very unfortunate for someone who wants to present the model. And so what I'll say is, I want to summarize this model, because what this model has is an origin of Neanderthals around 600,000 years ago with a lot of error, right? All these genetic estimates have a lot of error. A rapid spread of Neanderthals and a rapid diversification from Denisovans within a span of 30 or 50,000 years. So you have what happened in the earlier Middle Pleistocene was precisely the same as what we think happened in the late Pleistocene, except with different actors involved. You had a Neanderthal ancestral population that rapidly spread and diversified, that became sort of genetically differentiated, that spread into these different populations, and as a consequence, inhabited most of Eurasia. Well, that's a great model. I like it. Yeah, every archaic group that we've sampled interbred with others, often much more than we appreciate. Right? This is a really, really bottom line statement. But let's come back to this hybrid. Because if it is true that Neanderthals originated a long time ago, deep in the Middle Pleistocene, and that Denisovans originated also sort of deep in the Middle Pleistocene, and that these populations existed for a long time, that's convenient as an explanation for why we can tell the difference between them so readily in genetics, right? They are actually genetically pretty different. And the Neanderthals, by the way, are also genetically pretty differentiated. So we know that there's been time involved in their separation, in their diversification. But we also know that they hybridized with each other, with us, with other populations repeatedly. We know in evolutionary biology that these two facts are a contradiction. You cannot have diverging lineages become really different if there's lots of gene flow between them. So what explains this? How could it be that these populations that are spreading and able to exchange genes with each other, and who are cosmopolitan, as Milford says, able to spread effectively, and evidently able to transform over time within a region with some pretty substantial genetic turnover, how could these populations also maintain the kind of genetic difference that we see? Well, there are some ideas about this, right? One idea, often associated with Robin Dennell, is something I've written about, something others have written about, is sources and sinks. There are areas of the geography that have, sort of reproduce more, and areas of geography that are not really great for long-term habitation. And these establish clines that actually are long-lasting, even if hybridization happens a lot. I like the idea. We don't presently have great evidence about it. But I will turn for a minute. Human populations are invasive. Human populations are resilient, right? And these are somehow contradictory to each other. And I want to show you how this plays out in Africa. All right? African modern human populations, we today know a lot about their variation. We know a lot about their diversity today. And one of the things that we know is that there were groups as different as Neanderthals and Denisovans that interbred with today's African population ancestors. These are the African ghost populations. They existed as long ago as the early Middle Pleistocene and quite possibly earlier. Their sign is visible when we look at across the genome, when we look at different areas of the genome, and we look at how differentiated individuals in African populations are at different areas of the genome. So most of their genome has this sort of low level of differentiation, 500,000 years or something like that, but some parts have much greater differentiation. And those parts that have greater differentiation in their genetics are parts where some of the genetics has come from a diverged population. We also see the signs of this in today's genetic variability. Here are hunter-gatherer populations in Africa who have much more heterozygosity. That's much more genetic variation than anybody else. But in particular, if we look at a chart like this, I'm going to show you these PSMC plots, sorry. This is everybody outside of Africa today, sort of looks like this. What this is a chart that at different times in the past attempts to give a representation of how big the population was at that time in the past relative to today. So here's 1.0 scaled to today. Here's less, here's more. What you see is that everybody outside of Africa has this time when what looks like genetic variation was really, really low, right? And here it's in the time scale about 50,000 years ago. I don't want you to take the time scale super seriously. We don't really know if this is 100,000, if it was 30,000. But what we do know is it's shared. That signal is shared by everybody outside of Africa. That signal, it's here plotted in terms of effective population size, but what effective population size is a measure of inbreeding. How inbred are these people? Everybody outside of Africa is inbred in this time frame. However, when you look at hunter-gatherer populations in Africa, you see that that time frame where the French have this huge inbreeding, these hunter-gatherers don't, right? This is one of the big signals that you have some sort of bottleneck that has affected these populations outside of Africa now. Right? When people talk about the spread of humans out of Africa, that sort of thing, this is this data that supports it. In Africa, the hunter-gatherer populations don't have that strong signal. But if we extend this further back in time, here's the non-African populations and their big bottleneck-looking thing. Here's some African populations and their lack of a bottleneck. Here are Neanderthals and Denisovans. And you see that through most of their history, they look bottlenecked. And that's reality. If you look at these genomes, they look like they came from a tiny population. they look like a population that was highly inbred. If you look deep into their history, they start looking less inbred until finally they share most of the signature that we see in other living human populations. And that time frame is sometime long before these sequences that exist in the late Pleistocene, right? So sometime in the early middle Pleistocene, their history merges with ours. But after that, they're super inbred. The fact is that the population structure of this Eurasian population, Denisovans, Neanderthals, and anybody else that might have been there, that population structure was one that was divided into very small units, very small populations that interacted with each other less frequently. They were less outbred, right? Modern humans today are weird. I said it earlier, we're not weird. Actually, we are weird. We're weird in one way. We're weird in that today we're really, really outbred. Our recent ancestry in some populations was not that, was inbred, and in other populations was outbred. But our ancient history was much more inbred. All right, we know this from the fossil record. Split this. Oh yeah, I'll skip the ghost populations. There's some reasons to think there are ghost populations. We know this from the fossil record because when we look at early African modern humans, here, I want to show you this, the things that we look at and try to say, this is the origin of modern humans. No, this is the origin of modern humans. No, Jebel or Hood is the origin of modern humans. No, Homo is the origin of modern humans. Right? We all want to have the first modern humans. The fact is, when we look at these actual specimens, they're all really different from each other. And they don't share the same things with us. They're really diverse. And that diversity is a sign that Africa was also, in our early, late, and late middle Pleistocene time frame, genetically diversified into inbred populations that were actually quite different from each other. It's very popular today to talk about African multi-regional evolution. I like this, it's wonderful. But it's a very different type of picture than you would get if you took today's population structure and extended it back in time. In fact, the population structure at this time was quite more diversified than ours is, with less intermixture at a distance among populations than ours is. Which is one possible sign that you could have things that we haven't discovered yet, like ghost populations. Now, if I wasn't involved with discovering one of these, I might give this less emphasis. But the fact is that we have things in Africa, this is Homo naledi, that existed in the middle and later middle Pleistocene that look like they represent really old lineages that were there for a long time. So that kind of differentiation in Africa, which includes what people claim to be the ancestral population of modern humans, also includes really diverse things. So the diversity in the past was much greater than today in population terms, but the diversity in the past within populations was much less than today, vastly less. Okay, so where do I go with this? I told you I wasn't going to tell you who the ancestors of the Neanderthals were. All right, I'll tell you it's not Homo heidelbergensis. I'll just skip it. I'll tell you that these guys, right, Simidolis huesos, this incredibly wonderful sample, right, which genetically looks like an earlier branch of the Neanderthal lineage, right? Here's Neanderthal scenarios, and this is what people would have said 10 years ago. Simidolis huesos branches off from somewhere the common ancestor of this Neanderthal and African peoples. Today it's quite clear that Simidolis huesos branches off from an earlier branch of the Neanderthals, and that this all goes deeper in time. If that's true, if Simidolis huesos is fundamentally Neanderthal, we can ask, who's the ancestor of Neanderthals? Homo antecessor, right, is something that people talk about. From Atapuerca also, from Grundalina. And there's reasons for that. If you look at the specimens, they share a couple of features with Neanderthals. But that sharing of a couple of features is one sign, as we have seen, that can also be attributed to population mixture. if we actually have these populations that emerge, that become genetically diversified, and mix every so often, we will see this kind of increase over time. The Maurer sample, Arago sample, Atapuerca sample of Neanderthal traits if we look at one delineated part of the world, right? Our bias is that we've looked at one part of the world and we've asked, where does this trace back to? We recognize the bias that comes in from looking at one part of the world and tracing it back is that we'll find the features that characterize that part of the world in their earliest manifestation. All right, so yeah, this is one scenario. This is the Bermudez de Castro scenario of we've got the first Europeans that branch off, we've got Homo antecessor that branches off, and there's a mainstream, and Neanderthals branch off later, and I don't want to give them a name because, you know, maybe they're heidelbergensis or something. And erectus is branching off way earlier. I don't know. I don't like it. I'll tell you what I think. I think that a lot of people, when we talk about the origin of Neanderthals, are sharing assumptions that I don't share. One of those is that the European record has little to do with anywhere else. I actually don't share that assumption. I think the European record is very relevant to a lot of things. It doesn't mean it's the ancestor of everything. It means that it has relevance to understanding what is on other places. And the Neanderthals are no less a case of that. Neanderthal variability tells us about population structure broadly because it's a characteristic of the population structure broadly. Homo erectus is irrelevant. Because nobody talks about erectus and Neanderthal origins anymore. I don't share that assumption. And African Homo sapiens populations were more fluid than Eurasian Homo populations. That's something that a lot of people share as an assumption. I don't believe it. I don't believe that there's something different about Middle Pleistocene African populations in structure. I think that their structure is comparable to what we see with Neanderthals and Denisovans. Okay, so that being said, who are the ancestors of the Neanderthals? I think we have to bring a lot of these characters in, right? We know, for instance, that Asian Homo erectus, right, Zhoukouqian, is part, potentially, of a community that's contributing to the Denisovan population, this ghost population concept. We can't rule out these guys. They could be the ghost population. Here's a potential ancestor. This skull from Dhaka, from Ethiopia, an African, later lower Pleistocene skull, about 1 million years old. Where are we looking for the ancestors of Neanderthals? We're looking for a population that existed before 700,000 years ago. That population might have been like this. And this, the Buya skull from Eritrea. Again, similar age, similar geographic location. But we know that these later populations were among the ancestors of Neanderthals because we know that the gene flow that entered Neanderthals from other places came to them from populations like this. So we know some ancestors of Neanderthals. We don't know them all. This is the other million-year-old thing in Africa. I like this one. This is from Olograssile. It's a very small-brained, very primitive-looking thing. Is it among the ancestors of Neanderthals? Is it among the ancestors of Naledi? Is it one reason why Naledi is hard to place, right? These are all questions I think we have to have in mind. Okay, so that's where I want to stop.